In this last part, we are going to focus on patterns of inheritance. We actually have a long video in the this week's lab and talking about how do you use Punnett square and figure out the pattern of inheritance depending on the traits, depending on the alleles, right? And so we have a lot of exercise in the exercise 12, and basically use that information, right? And of the parents, the genetic information of phenotype and genotype of the parents, and figure out the offsprings, right? Genotype and phenotype. So strongly suggest you to look into that video to do the practice so that you can finish the practice in exercise 12. But for the lecture, we are going to focus more on the concepts and compare and contrast, and also this overview, the definitions and concepts. Okay, so that's why we start. This is a very similar, the same slides as in the lab, right? The overview of genetics. Again, we have just like when we talk about the genotypic sex and phenotypic sex, right? So here we have the similar type of distinguish. What the genotype is really the genetic makeup, right? Which gene or which allele you carries. And the phenotypes refers to the physical expression of the trait. Right? When we talk about the phenotypic sex, that was strictly on the external genitalia, right? But here, when we genetically talk about phenotype, it's really the physical expression of a particular trait, and we focus on those traits that can be decided by a single gene. Because if it's a physical expression of multiple gene interactions, it's just getting more and more complicated. And so for this course purpose, we focus on just the traits that can be decided by a single gene. Okay, so then we have this definition of autosomes and versus sex chromosomes. Basically, 23 pairs of chromosomes. We have 22 of them are autosomes, and then one pair of sex chromosomes. That's either XX or XY. Right, you can see the 22 pairs of chromosomes are here. They are different lengths and sizes, and their different genes localized into different chromosomes. So then when we talk about the genes, right, remember most of the genes are, most of the chromosomes are actually encode junk, right? Only like a very small percent, like less than 5% of that encodes genes, and the genetic, which basically codes for our all the proteins and some of the RNAs we make. Okay, so for each gene, right, because we have two chromosomes, we have a pair of chromosomes, they are homologous, right? We inherit one from our mom, one from our dad. And so we could have two copies of the same gene. There are variations of a single gene. So each variant then is called allele, right? So that basically they encode the same protein, but then they could be slightly different, right? Giving you slightly different makeup, genetic makeup, even it's for the same protein or same gene. And depending on the allele, so if it's allele that's dominant, that basically means even if you only have one copy of that allele, you are going to basically that trait that encodes by that allele is going to be expressed, right? It doesn't matter whether the other allele you have is the same allele or not. As long as you have one copy of that's going to be dominant, that you are going to express that trait. That's the definition of dominant allele. So that dominant allele always get expressed. And the recessive allele, on the other hand, means is a trait that encodes a trait that tends to be masked by the dominant allele. So you won't, if you have a recessive allele, if you only have one of those because everybody carries 2 alleles, right? Two chromosomes, 2 alleles. If you only carry 1 allele, then 1 recessive allele, it won't get expressed. Only if you carry both of the alleles you carry are recessive alleles, and basically the recessive variants of the gene, then the phenotypic trait is going to be expressed. So that's called recessive allele. Then that kind of lead us into this definition: homozygous versus heterozygous, right? Homozygous meaning a person with 2 identical alleles. Could be 2 identical dominant alleles, could be 2 identical recessive alleles, but as long as it's 2 identical alleles, that's homo meaning the same, right? Homozygous. And then if it's some person is heterozygous, then that means the 2 alleles it carries are different. So if we focus on the dominant allele, right? So the dominant allele, if you are homozygous for the dominant allele, right? Dominant allele are presented, we use capital letter for dominant allele. So if you carry or you are homozygous for this particular allele is widow's peak. The hairline here, it has widow's peak. If you carry the homozygous dominant allele, you present with widow's peak. If you are heterozygous with the widow's peak, so one allele is a dominant allele, the other is a recessive allele, you still present with the dominant allele phenotype, right? So that's why it's called a dominant allele. And on the other hand, the recessive allele, for that to be expressed, to see the phenotype, you need to be homozygous for that recessive allele. So then only when you have 2 copies, right, you are homozygous for this recessive allele, you are going to see the phenotype that associated with this allele. That will be a straight hairline. So this giving you an example. Right? So the recessive allele are presented, we usually write it as lowercase letter. We use lowercase letter to represent this is a recessive allele. So if you are given parents genotype, so this is from a mother with heterozygous with those 2 alleles, and a father with heterozygous with 2 alleles, then you can figure out what are the possible genotypes of the offspring. So we use that by Punnett square. In the lab, we went into details. How do you generate a Punnett square? Right? How do you then from the Punnett square, using the genetic information, genotypes, right, of the offspring, and figure out the phenotypes. At the same time, also you can get a percentage, because each, right, there are 4 different kind of combinations based on the based on the mother and father's genotypes. Because each of the gametes is going to carry only one of the alleles. So there's 4 different combinations. So each those squares represent 25% of chance that the offspring going to have this genotype, right? So then for this particular kind of parents genotypes, you're going to actually have 25% of the chance, right? 1/4 of the chance getting homozygous dominant alleles, and then 25% and plus 25%, right? You have 2 of those blocks. Out of 4, that will be 50% chance of getting heterozygous genotypes, and then 25% of the chance to get homozygous recessive alleles, right? So that's the genotype percentage. Then if you look at the phenotype percentage, you are getting 75% of the chance with the widow's peak, that will be the dominant phenotype. Only 25% of chance with the recessive phenotype. Right, so this is then how you use the Punnett square, figure out the genotype, then figure out the phenotype percentage, right, based on the parents' genetic makeup. So you end up with only 2 types of phenotypes, but then 3 types of possible genotypes. When you have, right, 1 dominant allele, 1 recessive allele, and if the dominant is complete, right, that means if you have a dominant allele, you are going to present with the dominant phenotype. So that's what we call a strict dominant recessive kind of relationship. But the real life situation is actually more complicated. Sometimes we have more than 1 dominant alleles, right? Sometimes if it's a dominant allele, but then it doesn't, it's not 100% dominant. So it's kind of half of the time is going to, so put it other way, if you have a heterozygous, then it's only you only end up expressing 50% of the protein, the protein that get expressed is only 50% of the amount of the homozygous dominant allele. So then the phenotype becomes intermediate, right? So it's become in between. And then in that case, then you are going to end up with 3 genotypes, 3 phenotypes, right? So we're going to go through that one by one, and I'm going to give you some examples. So those are, we're going to focus mainly on the first 3 types of pattern of inheritance. In the lab exercise 12, you actually are going to actually practice, right, with all 3 types of scenario. So for the details, you really need to do the exercise yourself, right? And but here, just to summarize that, right? If you have a strict dominant recessive inheritance pattern, and then you are going to have, like I said, 3 genotypes. Looking at the last figure, right? 3 genotypes, but 2 phenotypes, because the heterozygous is going to behave like a homozygous dominant. So that's what we call it a Mendelian inheritance, right? So throughout the high school, you actually take biology, you are familiar with that. And the dominant allele, the features dominant allele always expressed in the phenotype, and express, if you have 1 copy of the dominant allele and 2 copy of the dominant allele, the expression is the same, right? The phenotype is the same. When you have incomplete dominance, right, that's the one are going to have an intermediate phenotype, right? So none of the allele is completely, so if we still have 2 alleles, then none of them is acting like the dominant allele, like we already talked about. So if you have 2 heterozygous allele, right, the phenotype is intermediate between the homozygous dominant or recessive. Okay, the example here is a sickle cell trait. Okay, so your textbook actually have a explanation is this protein get expressed, right? Normally, if you are homozygous, for that sickle cell mutant protein, mutant gene, so you're going to have a dramatic phenotype. Almost all this red blood cells going to be weird shaped, right? Sickle shaped. And so then they tend to be clots, then you have problem carrying the oxygen, right? But then if you're heterozygous for that gene, and it's only 50% of the red blood cells going to be affected, and then so the intermediate, the deficiency in carrying oxygen is not going to be that prominent, and so all the phenotypic presentation going to be intermediate, right? Not dramatic as the sickle cell, homozygous for the sickle cell. So that's why you have an intermediate kind of phenotype. So this, instead of 2 phenotypes, you actually can see 3 phenotypes: a severe phenotype, an intermediate phenotype, and then a normal phenotype in terms of the sickle cell. Another example would be if you have a red flower, so that's in your lab exercise, and that's red flower is actually a incomplete dominant allele, and you have a white flower, right? That's the white flower allele, right? White coloring allele is also is a recessive. But then if you have heterozygous, right, a white flowering and a red flowering, you end up not with red, you end up with pink, right? Because the pigments then is half amount of in the red, so then presentation become pink. You end up with 3 phenotypes. So in other words, what if you don't know, right, whether this is the inheritance pattern is a strict dominant recessive or incomplete, but you observe, right, the phenotypes, and this trait is encoded by a single gene, but usually you can observe, for each kind of crossing, right, each kind of breeding or each kind of parents, then you actually can see 2 phenotypes. Then you only can see 2 phenotypes, right? So that means that must be a strict dominant recessive inheritance. But if you can see 3 phenotypes, and the phenotypes present as, one of them is the intermediate between the 2 more extreme phenotype, the chances are, well, that's a very good indication that this is the alleles behave like incomplete dominance. So the pound and square or everything, how do you figure out the genotype is still the same. It's just the phenotypes. Then the heterozygous individual is going to have intermediate phenotype. So the last type we're going to focus on is the co-dominant inheritance. That's the part the lab exercise devotes the most exercise into. A typical example, we actually are pretty familiar with this already. It is the ABO blood type. So your lab exercise focus on the ABO blood type to do the exercise. So that feature, so that what that means is now you have the 2 alleles may be equally dominant, right? So then for the ABO blood type, there's actually 3 alleles. Either you have A antigen, B antigen, or if you don't have any antigen, there are 3 alleles. So that's why it makes the genotypes and the phenotypes more complicated, because then you have multiple possibilities of the genotypes. But if you only have 2 alleles, let's see, we can think about what if a chicken only have the color, the feather, right? Now you have the red feather, you have the white feather. Okay, so then those the red feather are encoded by the red allele, right, and the white feather are encoding by another co-dominant allele, and they are both equally dominant. So if you're homozygous for each one of them, it's easy to understand, right? If you're homozygous for the red feathers, then the chicken will be red, having all red feathers. If the chicken's homozygous for the white feathers, then the feathers will be all white. But if the chicken has heterozygous for the white and red feather, what do you think the feather is going to be like? Well, if you end up with pink feather, that's incomplete dominance, right? Because that's intermediate. The co-dominance, you wouldn't have pink feather, because both allele are going to be expressed. So the chicken will end up with both red feather and white feather. So you're going to have like a chicken with both feathers, both colored feathers, not pink feathers, but both red and white. So that's an indication of the co-dominance inheritance, because then you actually see the expression of both alleles. ABO is another type, right? And so the three, how that's kind of giving you, so after we summarize in this way, this is different than the way we actually doing it in the lab exercise. Because now I'm really asking you the reverse question, right? If you see the phenotypes from particular, from inheritance pattern from a particular pair of two alleles, can you then tell based on the phenotype? Then you can tell whether the two alleles are strict dominant recessive, whether they are incomplete dominance, or whether they are co-dominant. I believe you can. Okay. So then the last one we're going to talk about is only one sentence, right? Polygenic inheritance. That means the trace is decided by multiple genes. And so there's interaction between the genes. And the genes we are talking about is usually on different chromosomes. So they're independent transmitted and inherited, basically. And then that's going to be much more complicated. Like I said, it's going to be beyond the scope of this course. But guess what? Most of our supermajority of the traits, right? Your height, most of the features, your height, your weight, whether you get certain disease or not, whether you get cancer or not, right? Most of the things we really care about, right, is actually transmitted, right, inherited, decided by multiple genes. So it's no strict, and plus then there's a lot of times interactions, and so it's not going to be as straightforward as the widow's peak. If your mom has this, your dad has that, then you're going to end up having this percentage of chance of getting this or that. Most of the especially disease, right, are decided by multiple genes. So they're called polygenic inheritance. Now we are going to move on to the sex-linked inheritance, and this we also talk about in fair length in the lab exercise 12. But then basically if the gene is on the sex chromosomes, right? Especially when we talk about this, is most of the genes on the sex chromosomes are on the X chromosomes. That's because X chromosomes are much bigger than the Y chromosomes. And also what's on the Y chromosomes, the genes on the Y chromosomes are predominantly, a supermajority of them are determining the male sex, so there are sex-linked traits, right? So then when we talk about the sex-linked traits, the sex-determining genes are on the Y chromosomes. So what's on the X chromosomes are really are a wide range of genes encoding not related to the sex determining the sex presentation. So and so then that's why when we talk about sex-linked inheritance, we mainly are talking about the X-linked inheritance. So there's two types of the traits, just like on the autosome, you have the dominant and you also have the recessive traits, right? Let's start with the X-linked recessive traits because that's the most commonly seen. And examples of that would be color blindness and hemophilia. Hemophilia is this blood clotting disease. Hemophilia A, there's different type of hemophilia, but then hemophilia A, we know is actually carried by a gene on the X chromosome, and its transmission is recessive. So usually it's presented in the male because the male only have one X chromosome. So if it carries that, if the only X chromosomes carry that recessive trace, then it's going to be presented, right? But then for female, because the female tends to have two copies, well, not tends to, but all the females have two copies of X chromosomes. If only one of them carries the recessive trace, then it's not going to be presented because it's going to be masked by the dominant dominant allele. So only the X chromosomes, only both of them are carrying the recessive allele, then you're going to see the phenotypes associated with that trait. And so that's a low probability, basically, because then that means it says that both parents would have to carry that, right? And especially then the father would present with that traits, right? Because the father only have one X chromosome, and that's the one have recessive allele going to be passed down to the daughter. So that's usually easy to be detected. If the father already have the disease, it's going to transmit it to then the offspring, the daughters, okay? Then when we talk about X-linked carriers, so that refer to the females that have only one copy of the recessive allele, because the presentation is going to be masked, and so it's only carried and looks if it's a disease gene, so this heterozygous carrier would have normal phenotype. So we have actually went through this in the lab, exercise 12. So I wouldn't repeat here. If you are having trouble with this, then listen to that recording. But basically, it's the same way if you are making a Punnett square, but the only difference is now you need to include the sex chromosome in the writing, not only the allele, because if it's a male, right, it's XY. So you want to actually include that sex chromosome in generating this Punnett square. So, but I do want to point here. So if you look at, right, if a male carries a copy of the X chromosome with the recessive allele, that's a disease allele, right? So the hemophilia A is this example we use here. And then this male is going to be affected. It's going to be hemophiliac, right? But then if it's a female that carries this X chromosomes, the same X chromosomes, right, with the recessive disease-causing allele, its expression going to be masked by a normal clotting allele that on the other X chromosomes. So this presentation, the presentation of the phenotype of this female is going to be normal clotting, but it is called carrier because it's going to carry that and it's going to actually pass that. So if this is a carrier female with an affected male, a normal male, then the carrier, the gene carried by the carrier female is going to be passed onto its one of its their daughters, right? But then it's also the daughter is still going to be carrier, it's going to behave normal, but the main affected person is going to be sons, right? The male offspring is going to have a 50% chance of getting that disease. So the X-linked recessive gene predominantly, right, affecting male offspring. Well, there are still X-linked dominant traits there, but it's relatively rare because then it's basically whenever you have it, you're going to be expressed. It doesn't matter whether it's in the male or females, right? But even in this case, the man still typically gets more severely affected, right? Because it doesn't have a normal X, normal X to kind of provide some kind of function, normal function. So in that case, right? So if the female present are heterozygous for the X-linked dominant traits, you will see the traits, but it tends to be less severe. And many times, if it's a severe condition, the X-linked dominant, the males with the X-linked dominant disorder are going to be spontaneously aborted because you won't be able to survive. Well, this is the last slide, right? We're going to end here, and we want to introduce this concept called penetrance. So this is looking at population-wise, right? So it's the percentage of the population with a particular genotype that exhibit the expected phenotype, right? So this is, again, not looking at the individual, but looking at the whole, population. Because some of the traits, right, based on the genetic makeup and what we know, whether it's a dominant and a recessive allele, it should all express. But maybe we see in the population then only express in 90% of the case or 80% of the cases. Then how do we square that? Well, that's actually indication that there are a lot of factors can influence the expression of this particular genotype. Right. Many of those factors include are environmental factors. So we know, how we behave the way we behave, how we get certain disease or not. It's going to be, the environmental factor going to have a huge influence on that, on the genetic traits, right? During the development, in the womb or growing, all the development either inside of the womb or outside of the womb, you could actually come across with multiple different chemicals, and some of them are considered teratogen or that can interfere with the phenotypic development, right? But also nutrition plays a big role. And so poor nutrition is going to have a negative effect on development. But also, alleles, we said, especially when we said cancer, we said some of the alleles, some of the genetic mutations you carry, or genetic, variation of the genes you carry, predispose you to development of certain cancer, but doesn't guarantee you are going to develop that cancer, right? It's just increase your risk. That's still depending on the environmental factors, but also could be the other genetic makeup of other genes you have, right? So that's all going to influence whether you are going to have a particular phenotype or not. So that's the concept of penetrance. Just be aware, right? In reality, most of the genes doesn't have 100% of penetrance. It's always going to be, an interplay between the environmental effects and the other genes effect on the expression of a particular traits. So that's it. And that's the concept you are going to deal with in terms of the heredity